Hi there, this is our second lecture of the second chapter of Robbins Basic Pathology. So in this lecture we will study about cell death and different mechanism of cellular death which involve necrosis and apoptosis. So in this lecture we will discuss necrosis mainly in detail. So as we discussed in the previous lecture that when cells are injured they die by different mechanism depending upon nature and severity of the insult, and these different mechanisms include mainly necrosis and apoptosis, and sometimes cellular death follows a method in which there is combination of both processes, necrosis and apoptosis, which is termed as necroptosis. So what is necrosis? Necrosis is basically accidental cell death when there is sudden and severe cellular injury due to ischemia, due to toxins, or due to various infections or trauma. The method of cellular death is necrosis, and in necrosis damage is of so extent that it cannot be repaired by cellular mechanisms and cell cannot survive. However, if the injury is less severe and there are chances that cellular mechanisms are somehow functional, then cell follows a way of programmed cellular death, which is termed as apoptosis. And programmed cellular death or apoptosis is regulated by different pathways and different genes. So it can be targeted through various drugs. So cellular death via apoptosis can be regulated by therapeutic approaches, while cellular death by necrosis can't be regulated. And one more thing is here, necrosis is always pathological, while apoptosis is pathological as well as physiological. In physiological cases of apoptosis are those in which normal cells are removed, such as in GIT cells at the top are shed off by apoptosis and new cell from the bottom replace them, as well as in developmental process the interdigital structures are removed by apoptosis. So apoptosis is a physiological as well as pathological process, while necrosis is always a pathological process. It is also a point to remember here that cellular functions are lost long before cell death occurs. So if cell death occurs after 30 minutes, cell function are lost, maybe lost even after a few minutes, 1 or 2 minutes or so on, as here is the example of myocardial cell. In myocardial cell, the contractile function is lost after 1 to 2 minutes of ischemia, but cell dies after 20 to 30 minutes. But the morphological features of the cellular death appear even after 2 to 3 hours. After 2 to 3 hours, we can see morphological changes in electron microscope. However, in order to see in light microscope, time gap may be 6 to 12 hours. So cellular death occurs immediately after cellular death does not occur immediately after the loss of after the ischemia. Instead, it takes some time. However, the contractile function is lost immediately in myocardial cells. So now we come toward the necrosis in detail. So necrosis is a form of cell death which occurs in accidental states, and it is always pathological. So what happens in necrosis? Cellular membranes are damaged, cellular enzyme leak out, and when cellular enzyme leak out, they not only digest the damaged cell but also they damage the surrounding cell. So it is always followed by an inflammatory reaction. However, inflammatory reaction does not appear in apoptosis, which is regulated or programmed cell death. In apoptosis, there is no leakage of cellular enzyme outside, so inflammatory reaction does not occur. But in necrosis, there is always inflammatory reaction because cellular content and cellular enzyme leak out and they damage the surrounding cell, and leukocytes are recruited to the site of damage. So biochemical mechanism of necrosis differ depending upon stimuli, and the mechanism may be the failure of energy generation due to damage to mitochondria, they may be due to damage to plasma membrane, or they may be due to damage to cellular lipid proteins and various enzymes, or damage induced by reactive oxygen species. So there are multiple causes of necrosis and they induce different mechanism of cellular injury. Moving on next, necrosis may occur in a part of tissue or entire tissue or even in entire organ. So when here we see that when there is a injury to specific tissue, this tissue specific enzymes are released out and they enter the circulation, and when they enter in circulation their level increases in blood, and which is a important diagnostic criteria for several important diseases. For example, cardiac muscle contain enzyme creatine kinase as well as contractile protein troponin. So when there is myocardial infarction, cardiac specific CK and protein troponin, cardiac troponin increases in blood, which is an important diagnostic criteria to diagnose the myocardial infarction. Similarly, hepatocyte contain transaminases as well as bile duct contains alkaline phosphatases, which are helpful to diagnose the damage to hepatocytes or liver. So whenever there is irreversible injury or necrosis of the specific tissue, tissue specific enzymes are released in blood, which help us to diagnose several clinical conditions. Now we come backward and discuss the morphology of necrotic tissue. So when it comes toward morphology, there are cytoplasmic changes as well as there are nuclear changes. When it comes to cytoplasm, there is increase in eosinophilia in cytoplasm. What's its reason? It is because when there is necrosis, the proteins are denatured. When the proteins are denatured, these denatured proteins bind tightly to the eosinophil or eosin. As a result, there is eosinophilia in the cell, as well as the basophilia of cytoplasm is lost due to loss of basophilic RNA in the cytoplasm. Moreover, the cytoplasmic appearance becomes homogeneous. The organelles inside the cytoplasm are damaged and they are also lost. This is the characteristic cytoplasmic features of necrosis. Moreover, the myelin bodies are more prominent in necrotic cell. What are these myelin bodies? These are basically accumulation of fat cell which increase in the cytoplasm of necrotic tissue. And by electron microscopy, you can characterize the discontinuities in plasma membrane and organelle membrane of necrotic tissue. Now we come toward the nuclear changes which happen in necrotic tissue. So what are these nuclear changes? First, the nuclear shrinks and increases in basophilia. This nuclear shrinkage and increase in basophilia is termed as pycnosis. Then it is this shrink the nucleus is followed by the shrink nucleus undergo fragmentation, which is called karyolysis, and then this all nuclear material undergo dissolution by DNase enzyme, and this is termed as karyolysis. So pycnosis, karyolysis and karyolysis are the different steps through which nuclear changes occur in necrotic cell. What happens? First, nuclear material shrinks and increases in basophilia, which is called pycnosis. Then it undergo fragmentation, which is called karyolysis. Then this fragmented nuclear material undergo dissolution by DNase activity of enzymes, which is called karyolysis. So after 1 to 2 days, a nucleus in that cell is completely dissolved and it disappears. So what is the fate of necrotic cell? Necrotic cell may persist for some days, but then they are digested by enzymes. When they are digested, they are replaced by myelin figures or fat bodies, and these fat bodies are further degraded into fatty acids, which combine with calcium and become calcified. So this is the ultimate fate of a necrotic cell. They may be replaced or eaten by phagocytes. Next, we come toward the different morphological patterns which may appear in necrosis, or simply say these are different types of necrosis. So necrosis has six main types, which includes coagulative necrosis, liquefied necrosis, gangrenous necrosis, caseous necrosis, fat necrosis, and fibrinoid necrosis. First, come toward coagulative necrosis. Coagulative necrosis is a specific type of necrosis in which the tissue texture is preserved. It is not lost. It is because when there is necrosis, not only the proteins, organ proteins are denatured, but also the enzyme, the digestive enzyme are also denatured. When there is loss of enzymatic activity, cellular texture is maintained as cells are not digested immediately. So in coagulative necrosis, initially firm texture of the organ is maintained due to denaturation of digestive enzymes, but later on the lymphocytes are recruited, and these lymphocytes release their enzyme and digest the tissue. So in the end, the organ or tissue is digested. So this is coagulative necrosis, and coagulative necrosis is the characteristic of infarcts occurring in major solid organs except brain, because brain follows liquefactive necrosis. So what is liquefactive necrosis? Liquefactive necrosis occurs mainly in bacterial, fungal, and viral infections. When there is infection, these microbes stimulate the lymphocyte, and lymphocytes are recruited immediately. When they are recruited immediately, they immediately start an inflammatory reaction, and they immediately liquefy the tissues. And it occurs in these infections, as well as central nervous system also follows liquefactive mode of necrosis. So in the liquefactive necrosis, in specifically bacterial infection, there is accumulation of pus at the site of infection or necrosis. Third comes gangrenous necrosis. It is not a distinct specific type of necrosis. Instead, it is a combination of coagulative necrosis and liquefactive necrosis, and it is specifically used for the limbs. When limbs are suffer from ischemia involving multiple layers of limb, then there is a coagulative necrosis initially due to loss of blood supply to the tissues. And then when it is initially coagulative necrosis, and initially the tissue texture is maintained and firm tissue composition is texture is maintained, but later on bacteria and the microbes invade, and there is an infection. When there is infection, it is followed by liquefactive necrosis. So it is a combination of coagulative necrosis and liquefactive necrosis, and when liquefaction starts, it is termed as wet gangrene. Next type is causous necrosis, and it is mostly seen in tuberculosis infection. It means a cheese-like appearance of the tissue. When there is causous necrosis, there is yellow-white appearance of the area of necrosis. And what happens here in this scenario, there is formation of a granuloma. What happens in granuloma, there is a central area of necrosis, which is followed by accumulation of macrophages. These macrophages enlarge and retain the shape of epithelioid cell. Even they form large giant cells, and then these macrophages layer is followed by a ring of lymphocytes, and then surroundingly there is maybe another layer of the surrounding cells, which are condensed and compressed due to these accumulation of lymphocyte and macrophages. So this is the case with causous necrosis, and causous necrosis is mostly seen in tuberculosis infection, that it leads to formation of granuloma. So it is also termed as granulomatous inflammation, which is a separate topic which comes under inflammation chapter. Next type is fat necrosis, which is specifically used for fat, abdominal fats. It is seen in emergency situation, which is called acute pancreatitis. What happens in this case? In this case, there is a release of pancreatic enzyme into the peritoneal cavity. When they are released in peritoneal cavity, they digest the fat found in peritoneum, and when the fat in peritoneum is digested, it combines with calcium, which is termed as fat saponification, and becomes calcified and forms local lions containing calcium combined with fat. So on histological examination, it appears in the form of shadowy outlines of necrotic fat cell, which are surrounded by basophilic calcium deposit. So it is typically fat necrosis, which is specific for abdominal fats. Next comes fibrinoid necrosis, and fibrinoid necrosis is a specific term used for necrosis of blood vessels. What happens here in fibrinoid necrosis? Antigen antibody complexes deposit into the walls of blood vessel, and they start an inflammatory reaction, which leads to the necrosis in the walls of blood vessel. It is fibrinoid necrosis, and it is characterized by bright pink amorphous appearance under microscope in HE staining. So it is a characteristic feature of arterial disease, which is called polyarteritis nodosa, which affects blood vessels. So fibrinoid necrosis is a term specifically for blood vessels. So what we see, we have studied here coagulative necrosis. It is the characteristics of solid organs, ischemia, or infarcts of solid organs. Liquefactive necrosis, it is a characteristic of infection, microbial infections, as well as CNS, necrosis of CNS. Gangrenous necrosis is a term specifically used for limbs, and it is a combination of coagulative necrosis and liquefactive necrosis. Causous necrosis, it is mainly seen in tuberculosis infection, and it is characterized by cheese-like appearance of the tissues. Then comes fat necrosis, which is a term specifically used for abdominal fats, and then comes fibrinoid necrosis, which is used specifically for blood vessel. It was today's lecture of necrosis, which was accidental cell death, which causes release of enzyme in cellular outside the cell and damages surrounding cell, and characterized by inflammation. In the next lecture, we will study about apoptosis and different mechanism of apoptosis. Thank you.